Okay, everyone, I have not uploaded the finished instructions to Canvas yet, so I'm just going to start with this, which also shows Lab 3, but this is Lab 4 instructions, so I will correct that before I upload this to Canvas. Everything is in the GitHub repository for this, so everything else is ready to go. I'm a little late getting this out to you because my RStudio has actually been tied up running something else for 24 hours. It is open now, so I can show you what to do. Um, we're going to start the same way that we always do by making sure we're in the proper project. So I moved specifically to a different project just to show this. So I've got a name up here that I just opened up this old project from when I taught POLS 3312 2 years ago, and I did all of my lecture slides in here. And so if you see something up here other than POLS 3316 spring 2026, you're going to want to change projects. And so how do you do that? One is you can open the project and search for it, and then you have to figure out where it's at. So this is why when you create these, you want to put them somewhere that you know you're going to be able to find them. And I have all of mine. I mean, I've gotten a little bit fancy here. I created a Windows library just for data projects that's out here, along with all of these other things. And I've got, different things. I've got 3 different kinds of data projects. But in your case, hopefully you put it in documents or maybe on the desktop in its own directory or its own folder. The other way, the easier way to get to it is if you've had it opened recently, you can just scroll down to recent projects, select it, and hit enter. I'm going to suggest that you want to do this every time because one of the things that happens when you do this is that it clears out some things from memory. It just makes things run a little smoother. If you actually switch projects, because what it does is it starts a whole new R session, and you can hit save when you leave it so that it doesn't lose any work. But I'm going to suggest it's usually a really good idea to do this even if you're already in the right one. Just good habit. It's also just a good habit because that way you're always working in the right one. So the next thing we're going to do is clear the console and the environment. And the way I do this is I use the little broom up here, clear objects from the workspace, and that clears everything, right? So I start with a completely empty environment, clearing the console. Mine is already clear, but I use clear console, and that actually gets rid of all of that stuff about what the R version is and everything. So if you need to send some of your console results to somebody because you need to help with an error or something, there's a little less there that way. It's just a little cleaner. It doesn't show the last 3 sessions. All right. So the next thing we've got to do, you don't need to do, well, let's see. Hopefully this will work without having to do anything else. I have some things that I need to send to Git. You won't have to do that. What we need to do is do the Git pull. So we're going to do that to update with the stuff that I've uploaded. And so to do that, the absolute easiest way to do that is use the green down arrow that says pull. So this green thing is an arrow. It points down, pull, because we're bringing stuff down to our computer from the cloud. Push, you don't need to worry about yet, and it probably won't work for you anyway on this repository, hopefully, because you should not have the credentials to do it, hopefully. But we're going to pull from the repository, um, get everything that we need. Good, that worked. So um, if you get any kind of an error, it's probably because there's a conflict between something out there on the repository and something that you have created on your own. So I suggested create something called like My Labs or My Data. I named mine Tom's Data and Tom's Labs. Those are where I keep the working files, where I actually go through and show you a demonstration. This Labs is actually where I've provided you files that you can refer to. So having a separate place for your work should prevent most errors. If you do get an error, there's code for dealing with it in Lab 3. I'm not going to repeat that here because some people were running that when they had no errors, and that was creating problems. Um, so, uh, so unless you have errors, don't worry about it. The system get pull, and the list, the RM list, those are just if you want to type instead of using the broom and using the green arrow. They're going to do exactly the same thing. So it's just redundant. You don't need to do those, and you definitely you don't want to put those in your lab script. How do you know you don't want to put those in your lab script? Well, we haven't created one yet. So the next step is we're going to create the script for this lab. And I'm going to go ahead, I didn't put this in the instructions, but it's always the first thing I do, which is to save it somewhere and go, and give it a name. So I'm bringing it out here to these labs that I've saved, and I'm naming it, actually, just to make sure it doesn't interfere with anything, I'm going to name it Tom's Lab 4. You can name it My Lab 4, whatever you want, but name it something reasonable and put it out in a place where it's dedicated to just your work, a folder that you've created. And you can create folders with new folder. Okay, so now we've started. The very first thing that I want to do is do the data loading. And so we did that in Lab 3, and I have to do it. I know that the Lab 3 stuff works on my computer, so I'm just going to go to Lab 3 and do that. But if you had errors, you still have to deal with them the same way that you dealt with them before. So if you had errors, you've got to go back to the what to do if there's an error thing, which is why I did not actually put that in this lab, because depending on whether you were able to successfully do this the way, not the easy way, but the more robust way, if you weren't able to do that, you won't be able to do it like I'm doing here. You have to use the other method. And so I'm not giving you two different methods. I'm letting you remember, did it work? Did I have to use the workaround? The workaround is in Canvas. The workaround is in Canvas. It's this thing. So if the lines of code that I just did don't work for you, there are 2 lines of code here that should work for you. And you can get to this. It's in the lab modules. It's also in an announcement. It's an announcement that's back about a week and a half, but it's the issue resolution with Gapminder and VDEM data. If you open that, those error fixes are there. But you just have to remember, you do one or the other. You don't need to do both. You may need to do the renaming step from Lab 3 in order to make this one work. I don't think you do. I think that I actually did that before I created the lab, the workaround. I think it's already got the renaming in it. But if you run into object not found errors, it's because you have to do that renaming step on this data frame, VDEM indices 2014. So the next thing we're going to do, because what we're going to do is compute correlations and covariances. This is actually pretty simple. It's not super hard to do this. We want to get the correlation and covariance for exports and polyarchy from VDEM, and then we're going to get the correlation and covariance for LifeExp and POP from Gapminder data. So those are just comments. I'm just copying and pasting them in. You don't have to comment exactly how I did, but a comment is just what it is. It's a signpost to yourself if you go back trying to figure out what you did, and it's a signpost to anybody who may read your code. So do it the way that makes sense to you. The first thing that I want to do or show you how to do, I've done this before, but I want to slow down and take a second to do it, is to get the variable names from those data frames so that we know what we're working with. And so we can do that with call names because the variables are column names. And so I'm going to do call names for Gapminder data. And I'm going to do call names for VDEM indices. And if you start typing and you just select the drop-down menu that pops up, you're not going to have to worry about typos or anything. Now, why did I do this? Well, it gives me the correct spelling and everything for all of these. So if you do this and this doesn't pop, this pops up with a bunch of things that say like V2X on the VDEM indices, it means that you didn't do the renaming. So you either need to do that or you have to adjust the code to use the correct, to use the ones that you have. But when I do this, I see things like the fact that LifeExp has a capital E. And that's really important because if I try to do the next part and I use just a lowercase e because all of these other things are, it won't work. And then for GDP per cap, it has a capital P. I could rename that. I know what they are, so I'm not going to. But it really helps to have the list of these. The next step then is, so all that is just housekeeping. But then the next thing we're going to do is we're going to build these functions step by step. And so there are two functions we're going to use: covariance, COV, and correlation, COR. So I'm going to use the question mark, which is the help thing, to bring up the help for these. And you can scroll through here. So it brought up, well, actually, these are so closely related, it brings up the same help file for both. And a lot of the things are going to be similar about them. So it tells me the usage. And so here's the usage if I want to do variance for a single variable. And we used that in the last lab. Now we're going to do covariance. And so the first thing I want to do is I want to do the covariance for the VDEM, and I want to assign it to COV1, and I want to do it for exports and polyarchy. Which I put up here. So correlation and covariance for exports and polyarchy from VDEM. So I'm going to find first the covariances and correlations and assign them to new object names. And why am I doing that? Well, because I want to use the object names later. I want to actually be able to use these. So I'm going to assign COV1 to, I want to make sure I do this the same every time. So I'm going to assign COV1 and then COV2 and then COR1 and COR2. So 1 is going to be for VDEM and 2 is going to be for Gapminder. see if I can actually type what I mean. So then COV2 and COR2 for Gapminder. So COV1, and if we look over here, it tells us that for covariance, I need X. I need the X. So, and then I need the Y. And if I don't specify a Y and I tell it just an X, I can tell it just an X for like the VDEM indices, and it'll return me every possible combination. I don't want to do that. So what I'm going to do is I do want to get variables from the VDEM indices. So that's the data frame name. And then remember, we use the $ operator to get the variable we want. And so as soon as I hit $, it actually brings me a list of the variables. This is one of the things that RStudio does for us. If we were working in base R, we would definitely have to go back up to the column names or down here to the column names and actually type every single letter. So this is useful. Then I'm going to put in the Y. So this is going to be the X variable, and then this will be my Y variable. So if you think about this in terms of cause and effect, what I'm actually saying here, if I were doing this where I was actually doing cause and effect, I would be saying that I believe that exports have a causal relationship, that they're the reason for increases in this polyarchy score of democracy that you may have heard about in some of your comparative politics or other classes. I don't actually believe that. I think it's probably the other way around, but there also probably is some feedback, but we won't get too far into that. So then the next thing is use. And I want to use something that's sort of a safe bet. I'm going to use something called pairwise complete observations. And what does that mean? And I'm going to hit return here. So what does that mean, corewise complete observations? Well, it means that there's a Y for every X. It's only going to consider the cases where there's both an X and a Y. And the biggest reason I use that really is that it helps prevent errors. You use some of the other methods and you can get errors if there's missing data. So the next thing is method equals, and we're supposed to pick one of Pearson, Kendall, and Spearman. So Pearson is the method that we use for finding correlation. And it's included for covariance just so that they'll match. But it's also the default. So if I come down here and I read method, it tells me Pearson is the default. So I don't actually have to type it. So I'm not going to. I'm going to move on. Except I'm not going to move on because I have made a typo. There is an unexpected comma. Well, there's an unexpected comma because I didn't type the function name. I just started typing after parentheses. There we go. So covariance. So it's COV1, I use the assignment operator, and then I have to use the COV function. So COV, and then the parentheses, and I find VDEM indices, $ exports, VDEM indices, $ polyarchy, use equals pairwise complete observations. The next thing I'm going to do is create COR1, and I'm going to be more careful this time. So if you did that last line, realize I did something wrong to start with. So you've got to go correct it if you were following along with me. This one I'm going to try not to do that. So exports, and then VDEM indices, VDEM indices polyarchy, and then I'm going to do the same thing. I'm going to do use equals, I'm going to come back here and hit enter so this is easier to read. Use equals pairwise complete obs. Obs is observations. And if you come down here further, there's even more explanation. So you get the usage, you get a short description of each argument, and then you get more of the detailed for the arguments. And then the last thing that you get is an example. And so you get some examples down here. So these help files are really useful once you learn how to read them. For some of them, well, I will show you when I get to Stargazer. So I'm done with VDEM, and now I'm going to do the Gapminder data. Look what I did up here. This doesn't matter because it's just a comment. But if I'd have done the same thing when I get ready to create COV2, so COV2 is going to be, I just used the function, so this is the name, this is a function, this is a different thing. So I'm not putting a 2 there. This is to create, I am assigning a value to this new object. And so now I want to use Gapminder data, and I said that we're going to use life expectancy and population, or life EXP, and I can select it. It will do the capitalization I need. Pop. So if I do this in this order, again, I'm kind of saying that I believe that increased life expectancy increases population. Hey, that one actually kind of makes sense, doesn't it? All right, so pairwise complete obs, just to make it a little more readable, I'm going to do that. One thing you should really take from this is you should really strive for readability. It makes it a lot easier. It just makes it easier on yourself. So when you are typing, put an extra row in between functions so that there's some white space. It makes it much easier to figure out what's going on. So now I'm creating this COR2, and I'm going to use the COR function, which is for correlation, and I'm going to do actually the exact same thing. So for the simple level, everything we do for everything we do for correlations is the same as what we do for covariances. So which you can kind of get if you come over here and you see that, you know, the format is the same. Many of the things that are the same. Again, I'm not going to put the method in. You can if you want to. The C here within the parentheses doesn't mean you should actually type C with parentheses. What it's telling you is it's using it the same way that we use the function. It's telling you you can pick one of those things. It's telling you this is the list of things that work. So when we combine some things to create a vector, we use that same C function. So that's what this is. It's a vector of the things that will work. The first one that will work is Pearson. The second one is Kendall. The third is Spearman. You don't actually use C. You pick one of these things from the vector. It's a little confusing, but if you start to think about it, it's kind of useful to do it that way. So the next thing that we're going to do, so we've got all of these. First thing I'm going to do is make sure they actually work. So I'm going to select all of them and run them. You might want to do it one line at a time. But by the time you're done, you should see them all filled out and then see some numbers out there. Don't look too closely at the numbers yet. You're going to spoil the surprise. So I'm going to close that down so we don't spoil the surprise. And then the next thing we're going to do is put these in a table. And we're going to make a really simple table by creating a data frame with the results. Which we'll do just like we did before. We're going to call it stats data. I think before we may have called it data frame. So I'm flipping back and forth a little bit here, but we're going to call it stats data. I think before we may have called it something like statistics table or statistics data. It doesn't matter what we name it as long as we know what it is. And so it's a data frame. And so these are just creating some titles. So we're calling data set, and we want to have the data set have the names of the two things we're using, but since these are just titles, we don't have to match anything. We're trying to make something that'll make the table look a little nicer. So we can actually, in parentheses, sort of spell it out in English. The next thing we want in that data frame, so the next row is going to be covariance equals, actually I think that's the next column, so COV1 and COV2, and then we're going to do a column called correlation that's going to have COR1 and COR2. So you want those to be in the same order because everything is by order. So when we create a vector like this, which is what we're doing, we're creating 3 vectors that are actually going to be rows in the data frame. Then what we want is to have 4 columns in the data frame. We want them to line up. We want them to be in the same order. So we put VDEM, it's 1 and 1. Gapminder is 2 and 2. So you gotta take care of the order. Now we want to actually look at the table. So if you did not install Stargazer, you can create a simple table from this by just typing stats data. And even if you did install Stargazer, you can still do that just to create a simple table that'll show up down here in the console. We can have a little more table control with Stargazer. If you installed it. If you did not install it and you want to try this, just go back and use install packages Stargazer. But I have to load the library first, and then I can give it, I can make this one pretty simple. This one is actually, I'll do a Stargazer example in a bit that will have more to it. But this one, I'm just going to make sure it does not include the summary. Because Stargazer's main purpose or most common use is to give a summary. And if we give a summary, it's going to actually repeat some of this stuff. So we don't want the summary, and we don't want the row names because we have column names set up. So we're going to leave the row names off. So that's false. And then we're going to do type equals text. The reason I'm doing type equals text is it will give us a nice little text file that we can copy over. So if I do stats data and run it, I have to actually create it first. So see, this is where running things one at a time helps. Okay, so I created the stats data. Then I can print the stats data, and here it is. And here's the big surprise, the big reveal. The covariance for Gapminder data is huge, and the covariance for VDEM is much smaller. But remember, covariance doesn't tell us the strength. So if you compare these two and you think, oh wow, there's much more. It's a much stronger relationship. But then when you go look at the correlation, which tells us strength, the correlation for VDEM is almost twice the correlation of the Gapminder stuff. So the Gapminder, even though it is a decent correlation, so it's weakish, but considering that we've got a limited number of years and some of the other stuff and how many other variables are involved, both of these correlations could be significant, and they are big enough effects. They could be important. But VDEM is twice as strong as the correlation. So the correlation between polyarchy and exports is twice as strong as the correlation between um, between life expectancy and population. And you'd expect a pretty strong, pretty strong life expectancy and population you would think would be pretty highly correlated. So that means this is actually probably pretty strong in some ways. But if we looked at the covariance, we'd come to the exact opposite conclusion. And the reason is the covariance isn't standardized at all. It's still useful. There are still reasons we do it. So we're going to do, at this point, then the Stargazer table. And it's probably, okay, it actually looks pretty good. If you've got this, it's going to look, it's not going to look that great. But because I have this pulled over where you can actually see it, and, feel free to move these things around. Just remember, if you move them around, like if you squash this down, you're not going to see some of the arrows and stuff. They're just going to disappear, but as soon as you expand it back out, they're back. So feel free to do that. A lot of times having more room to see what you're doing helps. I know some of you really like to squish this stuff down so that you can have the instructions and this on the same page, but it doesn't take that long to tab through. So that's Alt-Tab lets you, tab through everything. And I have a few things running because I'm recording and whatnot. But if you're working on one of these, if all you need is the lab document and maybe a web browser and then your RStudio, it's not as tough. So the next thing we're going to do is create something called a correlation matrix. We're going to create it as a table. We're going to create it as a simple graphic, and then a little nicer, and then a little nicer still. So we're going to create one table, 3 graphics. You're going to need 2 packages for this. The first one is dplyr. That's included in the tidyverse, so if you installed the tidyverse, it should already be there. If it's not there, all you got to do is do install packages dplyr, and it'll be there. The next one is new, so you probably don't have it. So we're not going to do this in the script. We're going to do this in the console because installation you do once. I don't know why I did dplyr, but why not? I'm going to cancel that. So installation you only do one time. Library is to load it. You do that every time, so it goes in the script. So I'm installing CorePlot, and then I'm going to go up here. I'm going to load the library. I'm also going to put in some comments first, creating a couple of correlation matrices. I'm going to call them matrices just to be fancy. Matrices is also correct. So I've installed those, and I've got the libraries. I'm not going to use these yet, but since I know I'm going to use them, I'm going to put them here and run them. It won't hurt to run the library more than once. In fact, dplyr is already loaded. I guess it's not. At the top, I thought I had put in tidyverse, but I guess I didn't. So they're both loaded now, but now we're going to go on to the next, which is the first thing we've got to do is we have to get rid of the non-numeric variables. If I try to run this, if I try to run a correlation matrix on, I'm going to show you what happens. If I try to run a correlation matrix on VDEM indices 2014, so don't do this. I'm going to show you an error. This is just to show you an error. If I try to do this, it's going to give me X must be numeric. So if you get X must be numeric and you're running one of these, it means you've got something in there that's not numeric. So how do we get rid of that? One of the easiest ways is to subset the data. So we're going to create a new piece of data, VDEM numeric. So I'm going to create something called VDEM numeric that is going to be VDEM indices. I'm going to start with VDEM indices 2014, and then I'm going to do something with it, and I'm going to use that pipe indicator. So this tells R, do whatever I'm about to tell you to VDEM indices 2014. So it tells R, whatever I'm about to tell you, go do it to that thing. And the thing I'm about to do is select, which is just what it sounds like. Select. We're just going to select specific columns in this case. And we're going to select specific columns where, which where just means where something happens. So it's actually pretty simple again. And the thing we're going to do is select the columns where it is numeric. So this is really just exactly what it sounds like. I'm telling it, I'm creating a new data frame named VDEM num, as in VDEM is going to have just numbers. I tell it, in order to do that, go out to VDEM indices 2014 out here and select, and then do the pipe, which is do the next function on it, or the next bunch of functions. And the function specifically is go out and select all the columns where the data is numeric. So actually, fairly sensible. This one's fairly plain English. Some of them not so much. There's a command in the tidyverse called mutate, and it has nothing to do with genetics. So it doesn't. I don't like it. Just because it's not plain English. But now that we've done that, we can create this correlation matrix. And you're going, hey, you haven't told us what that is yet. And there's a reason I haven't told you what that is, and that's because it's easier to just see it. And so again, I'm going to create a new object. This way I could use it multiple times. The object's going to be called core_mat, which is just, you don't have to use that. You can call it something else. But it just stands for correlation matrix. And then to create this correlation matrix, all I'm going to do is I'm going to tell it my X is the whole data frame. I'm not going to set a Y, which means it will interpret that as there is not a Y. I'm going to use the same thing I used before, which is pairwise complete observations, and I'm just doing that so that we don't get errors because of missing data. And then just because I can, I'm going to tell it method equals Pearson. I don't have to do that because Pearson is the default. When I did the example, I did it, so we're going to do it now. I'm going to run it. It's created that. It tells me that it's created this object that has 1 to 10 and 1 to 10, so it's a 10 by 10 matrix. It's created a 10 by 10 matrix. Why did it create a 10 by 10 matrix? Well, because the VDEM num has 10 variables. So the matrix is actually going to be every variable down the rows and every variable across the columns. And then if I look at that in the console by typing core_mat and running it, it's going to give me, and it's a little too big to fit right. But it's got, so here, this is the correlation between polyarchy and deliberative democracy. Very high correlation because they're two different measures of democracy, polyarchy and egalitarian democracy. So this is polyarchy versus deliberative democracy. I can come down here to deliberative democracy and polyarchy. And it's the same number, 0.97133852, 0.97133885. Some of them, for some reason, are rounding differently. But it's giving me all of the correlations, both directions, so I can look everyone up by row or column. We use these quite a bit. Okay, so we can also, if we want something that we can, so you can copy that and paste it into a Word document, but also if you want something with a little more control, and maybe you want to give it some titles or something. So we can use that Stargazer package again, and we can Stargazer correlation, that core mat object, and then I want to do the type equals text, which just gives me something that will transfer well to a Word document. It'll be plain text, it'll transfer well, but then I can give it a title. So I can say correlation matrix of VDAM 10 numeric variables. And then the last thing is I want to do align equals true. That'll give me a little bit neater table. And if I run that, it doesn't look very good in this. But if you copy all of this and paste it over to a Word file, which I just happen to have one open because here we have, well, maybe if I can find it. Here we have one. I'm going to come down to the bottom where the assignment is. Maybe. Okay. And it did not do what it should have done. Maybe I misunderstood what the align equals was. Let's try it again without the align equals and see what happens. See if I get a better result. If you get a really messy result because of following my directions on this, don't worry about it. That's my fault. Yeah, it still doesn't look right. Yeah, so I will do some investigating on that and figure out why it's, I don't use the text version very often. So admittedly, this is because I use a different version. I use the default, which is not intended to use with word processors. It's intended to use with typesetting software. Anyway, we don't need any of that for now anyway. And if it's a smaller table, it will come out neater. So I'm coming back up here where we left off. The next thing I want to do is create a simple correlogram or a plot. Correlogram. That is fancy word for plot of correlations. And there's a built-in R, it's either built in or it's part of the main packages of our thing called pairs that I can use. And all I have to do for pairs is, it's really similar to the correlation language. So I do pairs and I want VDEM num and I want use equals pairwise complete obs, just to make sure I don't get problems. And it produces this thing that looks like gobbledygook, but if you zoom into it, what it actually is this is a scattergram, a scatter plot of polyarchy versus liberal democracy. This is a scatter plot of polyarchy versus participatory democracy, deliberative democracy, egalitarian democracy. This is a scatter plot of liberal democracy versus polyarchy. Why aren't they the same? because this one has liberal democracy on the X and polyarchy on the Y. So if you look at them closely, they're flipped. Um, but it gives you every relationship, two ways of looking at it, um, and in scattergrams. So you can see like that participatory democracy and deliberative democracy have a really high, almost perfectly linear correlation, right? Um, the relationship between polyarchy and liberal democracy is a little more curved. So actually, there are some. If you're trying to get liberal democracy, there's some diminishing returns. Higher polyarchy doesn't give. It's still a positive relationship. It's just that as it goes up, something causes the increases in polyarchy to not make as big a difference in the liberal democracy score. So these are useful, right? Um, but also not terribly attractive, and you could use. So you've got some scales out here that aren't entirely clear what they mean, and you know, maybe it would be easier if you had actually rows and columns. And so we've got an alternative, which is to do the core plot thing. So we can do a really basic core plot diagram with this, and it still is going to be a little bit better. And we do that with just core plot and core mat. So the usage here is I just tell it core plot, that's the function, and I plug in what I want it to plot. And this one's actually pretty cool because um, it gives us polyarchy versus polyarchy. Obviously, those correlate at a 1 because they're literally the same thing. GDP per capita versus GDP per capita correlates at a 1 because they're the same thing. So it creates a table with dots that show us how things correlate. And a lot of these are really strong correlations. Some of them are pretty weak correlations. Some of them are very, very weak, like deliberative democracy versus exports and imports. But those are pretty weak. That's because there are lots of other more important variables like uh, economic variables that make a big difference, a bigger difference in exports. But if you controlled for all those economic variables, democracy really does increase those things. GDP, democracy, there are lots of other things that matter to GDP, like population. So you typically have bigger GDP if you have a bigger population. China has a huge GDP, even though its GDP per capita isn't as big. Um, so lots of complex things. So this doesn't give us a complete picture, but it still gives us some pretty important information that we can see graphically. What if we want to get something a little nicer? So we're going to get a slightly nicer using the same function. And I'm not going to explain this a whole lot because I think the best thing to do here is go and try to, play around with it. You're not going to break anything. If you get errors, you just erase it and do something different. Um, but go to the help file, which you can get to just through the question mark, and think about what the kind of things are you might want to do to show what you're trying to show. And if you decide to do that a little bit, with the homework assignment, I might give you a couple extra points if you can do it, and then you can write captions that tell me what you have done. So all this is really doing is adding some additional things, right? So I'm choosing black, but I could choose different colors. So just to do it, I'm going to use red. I'm not sure what that's going to look like, so I will change it back if it looks awful. And this is going to rotate the text a little bit. I'm going to put that final parentheses on the last on a line by itself. I think the reason that we put the final parentheses on the line by itself, if we have bunches of parentheses, then what we do is we put each closing parentheses on a line by itself with a different tab. And that way it makes it easier to count the closing parentheses and make sure that we closed everything. Um, so I'm going to run this. Uh, and okay, so it gave us red. First of all, this looks really messy, so I will zoom it out because it does look a lot better zoomed out. Um, so we've got red titles, and then blue is the basic color, and as the correlation improves, you get darker and darker blues until, these things are perfect correlations because it's population versus population, GDP versus GDP, and so these are essentially supposed to be black. They're very, very dark blue. And then we have a scale that shows. So this is actually a legend that shows what the numbers mean from -1 to 1 on correlation. And we don't have any that are anywhere near -1. We do have some that are a little bit negatively correlated, but nothing that's a huge negative correlation, which is not too surprising. I picked economic variables and democracy variables, and those are all positively correlated. Close that. I'm going to save this. We are almost done. So that's it, except for the assignment. If you can read this for yourself, you can stop watching. If you go read it for yourself and you need explanation, you can come back. So the assignment for Lab 4 is to find the correlation and covariance for your two variables of interest for the data set you intend to use for the semester project. Put these in the best table you can produce and copy it to a Word or other document. So you're going to find the first thing you're going to do is the very, very first thing we did up here, but you're going to do it with your variables from your data set, a covariance and a correlation. You only need one because you're only one data set, you need two variables. So I did exports and polyarchy. What if you have multiple variables? By all means, include those extra variables because when you do the semester project, you're actually going to need them all. Second thing though, if your data set has more than two variables, then the next thing is produce a correlation matrix in table and graphic format, place it in the Word or other document. Remember, you have to remove the non-numeric variables first, and there's an example of how to do that by creating a new data frame or you're going to get errors. You can do that with select where is numeric, that's fine. That won't work for categorical variables unless they're stored as numbers. So when I told you, and the reason is you can't run correlations or covariance on them anyway. You can't correlate apples and oranges. You also can't correlate oranges and GDP unless you're actually talking about the number of oranges. So when I told you the types of data sets to look for, this is one of the reasons. If you've got categorical variables, they have to be stored as numbers before you can do anything with them. And even then, correlations and covariances may not be as meaningful as you initially think. Like if you're doing some of those Likert score survey things, those correlations and covariances are probably not that accurate. They have some meaning, but they're not quite the same as if you had real continuous variables. Last thing on this, save the graphics or export them. And I guess this goes for the tables too. No screenshots, no cell phone pictures. These need to actually be properly taken out, exported or saved, and put into a document. And it needs to be one document. If I get 5 pictures and 2 text documents with tables and your lab R script, I'm gonna I'm going to reassign it to you. I'm going to give you a 0, reassign it, and say, turn this back in as a single document. Don't panic about the 0. You know, look for the reassignment and the comment, but I will do that. So be aware, it needs to go in a presentable document. And screenshots are not presentable. Um, Unless you want to do a lot of editing on them. If you're really good with photo editing software and you can make those screenshots look sharp, I guess that's fine. It's probably more work, though. Um, if your data set does not have more than two variables, so if you have a data set that just has, like if you're using the motor, I think it's the motor cars data set that has speed and braking distance, you only have two variables, so you can't do correlations or covariances. So in that case, um, well, you can do correlations and covariances. So you can do part one, and part two, but you can't do part three. You can't do the correlation matrix in table and graphic format. So in that case, for this lab, you can do your best thing is subset the VDEM indices 2014 differently than I did, so that your plot will be different. So that means subset it and take some of the variables out. That's one simple way. Second way is if you successfully installed the VDEM package, then you can go look at the code book, which is there's a link to it in lab 3, select 3 or 4 entirely different variables and create your own data frame from the VDEM package and use it. The third option is you can use the Gapminder data. If you've done everything, if you loaded the Gapminder data to start with, which you had to, because we already did some stuff with the Gapminder data, you can take the Gapminder data, take out the non-numeric data, and run it just like with the VDEM data. You need to produce a correlation matrix in table and graphic format. If you produce just the pairs, that's fine. You'll get full credit. If you produce one of the core plots and it looks nice, you'll and that means actually at the very least using some color. So if you use the basic core plot, that's also full credit. If you do something else with it, like some additional color or some other things, well, you're going to get that from time to time. I have done something that has made the machine mad at me. There we go. Okay. So if you want to do some other things like some labels and things like that, you can do that too. So there are a lot of things you can do with it. If you do something a little bit, if you do at least this, you'll probably get like half a point extra. If you decide to do something a little bit more complex, to show me that you've actually read the help file and figured something else out that you can do, that's worth a couple points. That is it for this lab. I'm going to post it. I know the video is pretty long. Actually doing the lab should be much shorter. Some of you may have only had to refer to specific parts of the video. Hopefully you don't have to watch the whole thing, but it's all here for you if you need the help. And you got a couple of weeks on this. All right, I will see you after spring break.